Thank you very much for inviting, and I'm very happy to be here, and I'm very happy to meet all of you guys virtually. I'm Nikolay Oskolkov from the National Bioinformatics Infrastructure Sweden, which is a bioinformatics platform of Lab. And what you see on my screen is sort of my symbolic or artistic view of today's meeting, because I feel like there are two groups of participants. Some of you are speaking more like mathematical and programming language, so you kind of have this natural science background, and some of you have a biological background. And those two groups, they speak dramatically different languages. Maybe oxidative stress and in situ transcriptomics is not necessarily understandable for people from mathematics. And I also assume that things like derivative-free optimization method and molecular dynamics computer simulation, that's not necessarily understandable for people with biological backgrounds. So I think it's important to keep in mind this diversity and try to find a common ground and use a simple language to kind of have a good collaboration. So I'm familiar with this kind of troubles because I actually did my PhD in theoretical physics a long time ago, but then I switched to life sciences and spent approximately the same amount of time in life sciences as in physics. So I am very well familiar with what Paulo was talking about, that he didn't understand anything from what Yuri Aulchenko was saying to him. Yeah, so that's a but it's a this cross-disciplinary research is not easy, and it's a topic for a completely different talk. So here today, I'm going to kind of challenge you guys a little bit and say, we are kind of at this meeting, we are trying to figure out whether or discuss how we can use machine learning in life sciences. Maybe we're already using it. Excellent. But basically, even people who are not familiar with machine learning, they know that machine learning needs a lot of data, and that might be a problem. So I'm actually going to ask you whether we have enough data in life sciences to actually run machine learning. I'm a little bit skeptic about it, and let's see whether you share my concerns. So what Nathaniel Street at the beginning mentioned, many people, I guess, are interested in genomics applications or machine learning applications in genomics. Yes, genomics, it's a huge direction in life sciences. People invest and develop this area a lot, and they spend years to build these huge genetic resources, human genetic resources, such as the Cancer Genome Atlas has thousands of samples with genetic information, methylation data, RNA sequence, and transcriptomics data. If you look at the UK Biobank, that's even greater. It has 500,000 of individuals. So 500,000 of individuals with genetic information, with comprehensive phenotypic information. That sounds like it's huge resources. So we are basically talking about hundreds of terabytes of disk space, or even petabytes of disk space. So this sounds like big. You can say, well, it's a big data. Let's try to use machine learning on this data. But you know what? This guy, his name is Mark McCarthy. And I'm a big fan of this guy. He's a professor at Oxford University in the UK, and his lab is one of the world leading in genetics of type 2 diabetes. And a few years ago, he was given a series of talks with the title, Now we have found over 400 genetic signals influencing type 2 diabetes. What on earth are we going to do with them? And it sounds a little bit like, doesn't sound very, very, very positive, does it? So Mark is a little bit skeptic and basically asking why, what are we going to do with these genetic signals that we couple to complex disease such as type 2 diabetes. So why can't we just use these huge genetics resources and take these signals. What you see on the left hand side is so-called Manhattan plot, where you can see many genes that were established to have something to do with type 2 diabetes. So why can't we take all of these genes and bring them to the clinics and try to take an individual genome and based on genetics, based on the DNA of this person, predict inclination of this person to type 2 diabetes or any other disease? Well, the simple answer is that it doesn't work. Uh, so, and the problem here is called the missing heritability problem. So basically, if you look at predictive power of genetics, of those genes coupled to type 2 diabetes. So this is a ROC curve on the left side. This is a predictive power of all these genetic signals. And it doesn't look great. Moreover, if you look at clinical factors, at prediction based on clinical factors, and clinical factors, it's a trivial information. It's like blood pressure, body temperature, body mass index, age, sex, and this trivial information turns out to predict type 2 diabetes so much better than genetics, and this is kind of frustrating. So why is it the case? What's the problem here? So the problem here is that the genetic variation data, genomics data, is high dimensional data. There are certainly multiple problems, but one of the computational problems that I am, as a computational person, is can have sort of competence to talk about is that it's genetic variation data is high dimensional data, which was already mentioned by Patrick, and he said that a few thousand sort of dimensions, that's a high dimensional data. So to give you sort of comparison in genomics in genetic variation data, we are talking about tens of millions of dimensions, and this is kind of hidden a little bit. People don't really emphasize that it's a high dimensional data, and it's sort of not everyone realizes, and not everyone, like Patrick, tries to say that that's a problem that actually leads to this the missing heritability issue. And so, what's the problem to work with high dimensional data? The problem is called the curse of dimensionality. So basically, in life sciences, when we do any statistical analysis, we typically work with matrices. And a matrix has two dimensions. And one dimension is the dimension of features, P. And the other dimension is a dimension of statistical observations or samples. So the features can be genes, proteins, genetic variants, and statistical observations, it's basically samples, or cells if you're working with cells. And features can be pixels, for example, if you work with image data. So depending on the ratio between P and N, you can roughly try to select one of those methods of statistical analysis. So basically, I would say, so if P is much greater than N, you probably should do something like Bayesian statistics. If P and N are comparable, well, preferably, of course, N should be greater than P. Then you're satisfying, you're fulfilling all the assumptions of frequency statistics with p-values, hypothesis testing, and power analysis. But if you happen to have huge data with N much greater than P, this is the case where you can and you should use machine and deep learning. You can go beyond linearities. You can go to some non-trivial types of analysis, non-linear analysis, not based on linear algebra. And if you look at sort of biological applications and biomedical applications, to my experience, they're sitting kind of here. So you people with biological background, you can think about what is N in your experiment is 5 to 6 mice, or maybe 20 mice, or maybe 100 mice. So your N is maybe 100 at most. And what is your P? So you're working with maybe gene expression, and then you have thousands of genes, right? So basically, you're probably here. So you see, you're very far from actually trying to do machine learning. Or, I mean, I can explain why you should not do machine learning if you have this type of limit, this type of data. So for example, you can run into this artifact. due to the curse of dimensionality. So if you run a simple logistic or linear regression, if you train a classifier, logistic or linear regression classifier, you want to establish a connection between your data, which is maybe genetic variation data X, and your phenotypic variable Y, maybe, or disease status, sick or healthy, or bad cells, good cells, and ugly cells. So these problems, linear regression problem, can be solved in closed form in this way. And here there is a so-called variance covariance matrix which needs to be inverted. And when you basically, to make a long story short, when you do this inverse, when you compute the inverse matrix, it happens to diverge in this limit. So it can be shown that and you never know how close to this divergence limit you are. Basically, if you are in this limit, in the high dimensional limit, so all your mathematics is very vulnerable. It can break very easily. So another example is this. It can be analytically derived that the variance or estimated in maximum likelihood way, for example, in this frequency statistic, traditional statistics way, is biased, and it becomes even more biased in high dimensional limit. So basically here you have explicitly, you have this interplay between N and P. And on the left side, you have the estimate. You have the estimate of the variance from maximum likelihood principle. On the right hand side, the sigma squared is a true variance. So your estimated variance happens to be more and more biased when you go to high dimensional space. So basically, there are a lot of pitfalls, analytical pitfalls that you may end up with if you sort of not really realizing that you're working with high dimensional data. And data can be big in size. So like genomics, the genetic variation data. So I was working with archived 6 terabyte files, genetic variation files, archived 6 terabyte files. They're huge. It doesn't mean that they're very useful. So indeed, like people, there is a lot of enthusiasm in life sciences area that we are sequencing more and more. We have sort of huge amounts of data in size or in volume. But if you sort of display genetic variation data in a matrix form, so here you have sort of this dimension n is very short. It has, well, if it's a whole genome sequencing data, then how many genomes can you sequence? Maybe 1,000. While this dimension is actually huge. We are talking about million dimensional space. So this is genetic variation data. But if you look at single cell RNA sequencing data, for example, it looks very different. Even though if you multiply N1 by P1, it will be equal to N2 multiplied by P2. It's 1 billion. I mean, those two data types, they're equally large in size or in volume, but you see that they look very, very differently. It's basically, it's much easier to analyze single cell RNA-seq data from statistical point of view. This is why I'm saying this is a big data. And genetic variation is unfortunately little data because you can't sort of use robust statistics without ending up with these divergences and the curse of dimensionality problem. So basically, I hope I delivered my message that genetic variation data is high dimensional. This is important to realize and be careful. And sort of no statistics or machine learning will or should be used directly without taking care, without dimensionality reduction, without regularization, without Bayesian statistics, for example. There are ways to overcome the curse of dimensionality, but you should kind of remember about it. But still, can you use, so far I've been a little bit pessimistic, but still, can you use machine learning on genomics data? So it turns out that actually there is a straightforward way if you don't consider one individual or genome from one individual as a statistical observation. But if you chop your genome, if you slice your genome into pieces, then you have millions or billions of these small sequences. And if you consider one sequence as one statistical observation, then you should do something like deep learning, and because you're really you really have a grotesque, huge sample size with millions of billions of statistical observations. So basically here it's just an illustration how you can use this approach on real world ancient DNA uh, research. So basically, if you have modern sequences from modern individual, and you have sequences from ancient individual, they're a little bit different. In terms of there is a gemination pattern at the ends of the sequences, typically present for ancient sequences, and you can sort of feed. You can use this as a training data set. You can feed these two types. You label your sequences 0, 1, 0, 1. You feed it into one-dimensional convolutional neural network, for example, and you learn these k-mer patterns that can discriminate between ancient and modern sequences, because that's a very, very big problem in ancient DNA research, problem of contamination. They want to sequence Neanderthals, but if they in parallel sequence a bunch of modern human sequences, because modern humans, they drop their DNA in the sample. So, and we don't want to sequence modern DNA. We want to kind of separate ancient and human reads. But in the same way, you can sort of detect functional regions in genome basically by providing sequences from functional and non-functional regions, for example, and doing this classification approach in this way. Because here, n equal to 1 is a big data. So one genome is a big data if you slice it. I hope I delivered this message. But there is an area where you don't need to think about dimensionality that much, and this is a single cell omics area. Basically, in single cell, it becomes almost a routine to sequence millions of cells, and then you have millions of statistical observations. So here you have a luxury problem. You don't. So typically in machine learning, people worry about overfitting, a lack of generalizability. You overfit, it works here now, but if you bring it to another sample, it doesn't predict. Like the case of the missing heritability, that's a typical overfitting problem. But here for single cell area, you actually have a luxury problem. You should watch for underfitting to get the most out of your data, and this is a paradise for machine and deep learning. This area in life sciences, and very popular. We and other people they use autoencoders for single cell omics. It's an unsupervised neural network which has this bottleneck in the middle. So if you kind of reconstruct your input and the output, you and manage to compress your data down to just a few dimensions, and it means you learn something fundamental about your data. So you can sort of reduce dimensions dramatically in this way. So this is a very popular architecture in single cell omics. And it's an unsupervised neural network, and this is relevant because single cell omics, it's predominantly an unsupervised field. So people want to discover cell populations in an unsupervised way, in an unbiased way. And how does it work? So for example, just to give you a quick example, so this is a principal component analysis demonstrating gene expression cells in terms of gene expression. And one point here is one cell, and different colors correspond to different cell populations. And you see that all these cell populations, they overlap heavily. But if you use some nonlinear approach, so PCA is sort of linear thing, but if you want to use, for example, autoencoder, then you can sort of separate the cell populations a little bit better. You can do it even better with another beautiful dimension reduction technique called TSNE. And so, but if you sort of, TSNE is usually run on PCA. So you do some PCA pre-processing before fitting it into TSNE, and you can do it better with autoencoder. So basically what I want to say is that if you process your data with autoencoder, you can, so there are multiple examples actually where you get a better resolution. So you can sort of get this cluster, purple cluster, more sharp. So here it's this cluster is quite distinguishable. compared to this picture. This is a sort of a benefit of using machine learning and deep learning approaches in single cell omics for data processing, for example, not really for visualization. Another thing that you can use neural networks for data integration. So if you have like this is an example from SciSec technology, which delivers two types of biological information. It's transcriptomics and proteomics level of information, and you can sort of combine those two types of information in merge them in a hidden layer of a neural network, and get again this cluster, this purple cluster. It becomes more and more transparent now when you combine your RNA-seq data with transcriptomics. This is very easy for clustering. So here you sort of don't need to do much work to try to separate the purple cluster. So that's just a quick demonstration, and I hope you got the idea that single cell array is very promising for machine learning approaches. And so here, just to summarize, a very last slide, just to summarize some directions in life sciences where you look promising, at least to me, for applying machine learning. As I mentioned, it's single cell and genomics, not genetic variation really, because genetic variation is a high dimensional data. But if you use actually the sequences, then you can do a lot of machine learning in genomics area. Two other directions are microscopy imaging and metagenomics. So microscopy imaging is not a surprise. I'm not talking about it at all because even people who don't do AI and machine learning, they know that AI usually has something to do with images. So yes, we have automated microscopy imaging that delivers millions of imaging. That's a paradise for UNet, CNNs, and Mascar CNN. So you can do image classification, you can do instance segmentation, you can do object detection, you can do lots of cool things. But this is nothing new for AI people. You do it basically for breakfast. But what is less trivial thing is this metagenomics. I think what the previous the Kemal was talking about, microbiome research. That's actually quite a low dimensional data. While we have tons of samples actually from in metagenomics area, we have tons of samples, and this data is not really high dimensional, so very promising and potentially very interesting for applying machine learning methods. And I know that they do a lot of machine learning and metagenomics research currently. So basically here I would like to stop and say that I'm from the National Bioinformatics Infrastructure Sweden. Thank you all my colleagues and all the foundations, and I thank you for your attention.